Thank you very much, Lilian, for your presentation. I did listen you. And well, I thank you, everyone that is here today. And thank you for the opportunity to give you this presentation. And well, as Lilian said, this presentation has the objective of talk about in situ measurements of chlorophyll-a concentration, and in particular, focusing on how to manage the different types of methods that are being used in intercomparison studies. This is because the primary objective of these seminars is the work that many of us are doing in the NANO Global Project, which will integrate the observations that each country is doing around the world. Now, I will begin in this presentation talking a brief introduction about chlorophyll-a, phytoplankton biomass. I will follow with methods that are being used for the measurement of in situ chlorophyll-a concentration. I will talk a little bit about the measurement of other pigments and about which of these methods are comparable, what we can do about that, and in particular focusing on the working in networks. Okay. The first question that we have to keep in mind is why to measure chlorophyll-a. The most important thing is that chlorophyll-a is a proxy for phytoplankton biomass. And changes in phytoplankton populations may impact fish and other marine life, which can affect economic productivity, food availability. This is important also to decision makers that can use this information as an indicator to understand the health and productivity of marine ecosystems that will depend on phytoplankton. And finally, marine primary productivity is an important agent in the global cycling of carbon dioxide. That is a major greenhouse gas, and variations in the concentration of the ocean's phytoplankton biomass can therefore explain trends in the global carbon budget. The remote sensing of chlorophyll-a has been a valuable tool to analyze temporal and global spatial trends. Something about that, Milton Campbell presentation, he talked about that. However, in situ measurements are more precise and allow the evaluation of trends not only at the surface, but also in the entire water column. For this reason, the objectives of many of studies, including those of the NANO Global Network, is the integration of in situ and remote sensing measurements. What is chlorophyll? Chlorophyll is a greenish pigment which has a porphyrin ring with a central magnesium that is indicated here in this diagram. And it also has the characteristic of having, in some case, an hydrophobic chain, the phytyl, that is shown here below. There are several kinds of chlorophylls: chlorophyll-a, chlorophyll-b, C, and D. Their molecular structure varies by one or several side chain substitutions that here are indicated with the red letters, R1, R2, R3. The R4 here is the phytyl chain. For example, chlorophyll-a, B, and D have the R4 or phytyl chain. However, chlorophyll-c does not. And well, the most abundant chlorophyll is chlorophyll-a. And it is considered, as I previously commented to you, it's a proxy for phytoplankton biomass. And it is considered as the major, as an universal pigment, because it is present in almost all microalgae, excepting the cyanobacteria Prochlorococcus, which contains divinyl chlorophyll-a. These pigments are responsible of absorbing light, which energy is necessary for photosynthesis. An important characteristic of chlorophyll A in particular is that it has two wavelengths of maximum absorption that I am indicating here in the blue and in the red parts of the electromagnetic spectrum. It is one around 440 nanometers and the other around 670 nanometers. And also, they have chlorophyll has the particularity of, or the characteristics of emission of fluorescence. Fluorescence emissions is around 675 nanometers, or in the red part of the electromagnetic spectrum. These characteristics are some that are used for the determination of the concentration of chlorophyll in water samples. And now the main thing, or the main point of this talk, how to measure chlorophyll A concentration. And it will depend on the instrument we have. We can measure concentration of chlorophyll using a spectrophotometer, which is based in the light absorption by this pigment, by a fluorimeter, that what is based in the fluorescence emission by chlorophyll, and also high performance liquid chromatography. I will talk a little bit about each of these methods. Well, all methods require the pigment extraction using some solvents. The most commonly solvents used are 90% acetone, 100% acetone, or even methanol. The general procedure to be followed when we want to measure pigments, the chlorophyll A concentration, is first the need for filtration of the sample. This filtration should be done using GFF filters, which have a pore size of around 0.7 micrometers. Then we have to storage the filter in aluminum foil, and this filter will be stored in liquid nitrogen, or when liquid nitrogen is not available, we can use ultra-cold freezers. When samples are going to be measured, there is the need now of the extraction of chlorophyll using a known volume of solvent. After 24 hours in freezer, the extraction is complete. The sample is then is centrifuged, and then the sample or the extract is ready for go to some of the instruments I mentioned before. When we are going to use a spectrophotometer, we have to take in mind that this instrument, what it does is measure light absorbed by a solution. In this case, the solution is the extract we did of the pigment of chlorophyll A. This extract is added in a cuvette that will be, or it can be from 1 centimeters or even 10 centimeters longitude. And the absorbance inside by the sample is measured and detected by the equipment. This is an example of how can be the absorbance of light by chlorophyll A, and it is also shown an example for chlorophyll B. Here we can see that chlorophyll A has the maximum in the red, around 663 nanometers, while, for example, chlorophyll B has a maximum around 646 nanometers. This peak or absorption peak of the wavelength of the absorption peak will vary according to solvent. In this example here, I have one that, an extract that was done using 90% acetone. After measuring the absorbance by the sample, we have to apply some equations to go to chlorophyll-a concentration per se. One of them of these equations are those of Parsons and Strickland that are based on the use of the solvent 90% acetone. You can see that we will take in account the absorbance of the sample at 665 nanometers, at 630 nanometers, and 645 nanometers. Another proposal is that of UNESCO, using also acetone, 90% acetone, where the wavelengths are 663, 645, and 630. And finally, there is the equation of Jeffrey and Humphrey, proposed in 1975, where they use almost the same wavelengths, all of them in the red, as you can see, 663, 647, and 630. Well, the measurement of absorbance require a blank using the solvent, the pure solvent, and also a correction for the absorption at 750 nanometers. These procedures are fully described in Jeffrey, the book of Phytoplankton Pigments in Oceanography in the 1997. But here I am focusing on the main points to give you to understand why, which equation we can use if we are working as a network, which is the more recommended if we have spectrophotometers to measure chlorophyll-a concentration. And the equation of Jeffrey and Humphrey is one of the most used and the most recommended by even by SCORE, UNESCO, and other international agencies. After having this constant of this value here of C, we will take in account the volume of acetone used, is the small v here, the volume of sample filtered. We can filter half a liter, 1 liter, 2 liters. It will depend on the concentration of phytoplankton in our sample. And also we have to take in account the light path of cuvette because it will influence the absorption by the sample. After going to this equation, we will have chlorophyll-a concentration in milligrams per cubic meter. There is one point that have to be taken also in account when we are measuring chlorophyll-a concentration, not only using spectrophotometers, but also with other methods. Yeah, this is pigments or even some other pigments that can be present in the sample and can interfere with our measurement of chlorophyll-a. Pigments have an absorption peak near that of chlorophyll-a. And for this reason, it was recommended in 1967 by Lorenzen, the acidification method. It requires that after measuring the absorbance of the original sample, we add 2 drops of chlorhydric acid, 10% chlorhydric acid, and we will read the absorbance before and after the addition of these 2 drops. New equations were also proposed to integrate these acidification methods. They are also based on the absorbance in the red part of the spectrum. Here you can see this is 664 and 665 nanometers. The letter A indicate after the addition of chlorhydric acid, and the letter B indicate before the acidification of the sample. We will apply 2 equations. The first one using values after acidification, and the equation here that we consider after and before acidification. At the end, we will have chlorophyll-a concentration, you considering also the same points mentioned before, the volume of acetone here, the volume of the sample, and the longitude of the cuvette. And we can also have an estimation of pigment concentrations using the same, almost the same equation, but inserting the value of phi that I put here as phi, that comes from this equation below. Always is necessary the blank and the absorbance 750 nanometers, or correct these absorbance here for this one below. Well, now after making the description of the general considerations for the measurement of chlorophyll using spectrophotometers, I will talk about fluorometer. Fluorescence by chlorophyll-a, as I previously told you, is in the red of the spectromagnetic spectrum, and this fluorescence is excited by blue light. That means that a fluorometer, we have an excitation filter centered in the blue part of the spectrum, as it is indicated here, and we will have an emission filter that will detect the fluorescence by the sample. The quantity of the intensity of the fluorescence will be proportional to chlorophyll-a concentration. And the fluorometer measurement, or the measurement of chlorophyll-a using fluorometer, was introduced by Hansen in 1963. It is 2 or 3 orders of magnitude more sensitive than spectrophotometric methods. For this reason, it is more appropriate for oligotrophic waters. It can measure even concentrations below 0.1 milligrams per cubic meter. And as with the spectrophotometers, we also have, we can also use the acidification method or the acidification correction to consider the influence of pigments in our samples. Well, what is the problem with fluorescence, and what is the problem again with pigments? And also, when we are talking about fluorescence, we have to also to consider chlorophyll B. In this figure that I took from the book of Jeffrey, I forgot to put the indication of the source of the figure, and we can see that here, these are the wavelength or the absorption maximum of chlorophyll-a and also chlorophyll C2, and it's also shown the chlorophyll B maximum absorption wavelengths. And in this side of the graph, we will have the fluorescence peaks of these pigments. You will see that here I insert a red mark here to indicate that filters used in fluorometers usually average all of these wavelengths. That means that they are exciting not only chlorophyll-a or this blue light or the blue filter. It's not only exciting chlorophyll-a, but also chlorophyll C and chlorophyll B. In this side, when we are measuring fluorescence emission, this is the longitude of the filter, and that means that when the filter or the equipment is measuring fluorescence emission, it is not only detecting the influence of the emission by chlorophyll-a, but also by chlorophyll B. Here below, we have the same for pigments, and you will see, for example, that that the maximum emission of chlorophyll B coincides with the maximum emission of chlorophyll pigment B, and the maximum emission of chlorophyll A, it's almost at the same wavelength, the maximum emission by field pigment. That means that the wavelength width of the filter is a problem when our sample contains not only chlorophyll A, but also contains chlorophyll B and field pigments. In order to solve this problem, there is the acidification method implemented to correct for these chlorophyll B and field pigments in samples. However, in 1994, Walshmeyer introduced the use of new lamps and field excitation and emission lamps in order to be more specific in the wavelength, the detection wavelength. That means, for example, this line shows how is the emission of other lamps and filters, and here is the emission or sensitivity of the filters used by the Walshmeyer method. And you can see that it's very, very specific. That means that we can detect chlorophyll A with no interference from chlorophyll B or field pigment. When we use this Walshmeyer method, there is no need for acidification. This method is implemented in some specific types of fluorometers. Not all fluorometers have these lamps. Well, another point that I will not mention, I did not mention with spectrophotometers, but it is necessary when using fluorometers is calibration, because we have to compare fluorescence reading against chlorophyll A standards of known concentrations to establish a correction factor for the fluorescence readings the equipment drew. Laboratories sell these standards, for example, Sigma Chemical. Calibration should also consider acidification. It is good that previously know the range of variability of chlorophyll in our study sites in order to prepare our calibration information considering this range of chlorophyll observed in the field. And it is recommended that at least 5 points should be used to do that. Well, there are many details that have to be taken in account in all these steps. I recommend you to look for the oceanoptic protocols for satellite ocean color sensor validation in chapter 17. There is a really detailed description of the steps necessary to give or to do a good calibration of fluorimeters. And also in the book of Jeffrey of 1997. Well, we also have to apply an equations to have chlorophyll A in milligrams per cubic meters. I am here showing you only the one that use the Walshmeyer method that requires this constant, that is the fluorescence sensitivity coefficient, the one that we obtain doing the calibration. F0 is the fluorescence response measured by the equipment. Again, the volume of acetone and the volume of water filtered. And finally, the most modern method is that of HPLC. This method was introduced by Wright in 1991. It is the most accurate method for chlorophyll A concentration measurement, and it also allows to measure other pigments concentration. It is much more complicated and with much more details than the previous two methods that I talked to you about. These and other points and raised that many laboratories do not have HPLC, or even that other laboratories prefer and can pay for have their samples analyzed in some standard laboratory. And the main attractive of HPLC is its accuracy for chlorophyll-a measurement, and also the detection of these other pigments. Some of them are chemotaxonomic marker of some phytoplankton groups. It means that using HPLC, we can not only have information on the concentration of chlorophyll in some sample of water, but also we can have information about phytoplankton groups that are present in our sample. Basically, this is the general structure of an HPLC. It contains a column, a chromatographic column. It also requires the use of some solvent, in the other methods to extract pigments. And there is also the need to inject the sample, as I am indicating here. The sample will interact with the solvent or one or more solvents. The sample will pass through the HPLC column, the chromatographic column, that will separate pigments according to their molecular weight. And finally, this information will be captured by a PCE or data acquisition system. Well, I consider important to mention that pigments, which pigments can be measured by HPLC. This is a list of all of them. You can see there are many pigments, and chlorophylls are at the top of the list. Here is chlorophyll A, B, C. Here is divinyl chlorophyll A, that characterizes of Prochlorococcus, as I mentioned, and other carotenes and other xanthophylls and even biliproteins. Well, here we have the groups, main phytoplankton groups in the ocean, and we can see that some pigments are specific for some groups. For example, divinyl chlorophyll A and divinyl chlorophyll B are specific for Prochlorphytes or Prochlorococcus. Alloxanthin is specific for cryptophytes. Peridinin is specific for some dinoflagellates, and prasinozanthin is specific for prasinophytes. That means that the presence of these pigments in some sample indicates that these groups are in the water sample. Other pigments are not exclusive for some groups. They are shared among groups, but they also have a chemotaxonomic value. The final result of the HPLC analysis is a chromatogram. Here I am showing you an example where you can see that pigments are separated according to their rotation time in minutes. That means that, for example, this is chlorophyll-a here. Here we have the signal of chlorophyll B. Another example here is peridinin, that chemotaxonomic marker for some dinoflagellates, and so on. It implies that the information given by the chromatogram have to be compared also for a calibration, or also by a previous calibration of the equipment that needs the use of standards, as I also mentioned, for fluorometers. But in the case of fluorometer, the standard was only chlorophyll-a. Here with HPLC, if we want to measure the concentration or detect the concentration of zeaxanthin or fucoxanthin or peridinin in a sample, we have to acquire the standard for each of these pigments. Well, many methods, there are many methods to determine the concentration of these pigments in a sample. I recommend you to read the 5th CWIFS HPLC analysis round-robin experiment report. It's a report of NASA. It has the code CHAIR number 5. It was published in 2012, where the main or the most used methods for HPLC analysis of pigments were compared. From this analysis, this publication and others, the most recommended method, the most accurate method, is that of Van Huykelen and Thomas, first described in 2001, and then described in this same 5th CWIFS HPLC round robin by Thomas in 2012. Basically, one of the main differences among methods are in the column used. It can be a C8 or C18. The difference will give that the separation of some pigments some pigments are best separated by one or other column. And also another difference among methods is about solvent usage. Some methods use 2 solvents, others use 3. For example, the method of Thomas uses a C8 column and 3 solvents for pigment separation in the chromatographic column. Well, and finally, some points to be considered are, well, consider the comparison among methods. I will come back to the title of or the objective of this talk, that was how to integrate available method and about intercomparison studies. Well, spectrophotometers has the method that uses spectrophotometers have a low sensitivity. They are not recommended for very oligotrophic waters. Its precision is high, but when chlorophyll-a concentration is high, coastal waters, but also if degradation products are in low concentration, for example, the presence of field pigments. However, the cost of this equipment is relatively low. I am talking about around $30,000. Well, fluorometers, they have high sensitivity, as I previously mentioned. It can be used in very oligotrophic waters. Their precision is high if the degradation products are low. However, as I mentioned, also it will depend on the lamp and the filter used, taking, for example, the method of Walsh-Meyer. And the cost is relatively low also. And then comes HPLC. It's high sensitivity, high precision. However, the cost of this equipment is also high. And it is also high, the cost of the standards that have to be used to calibrate the instrument. That means that HPLC is an instrument that is not really spread in many countries. It is not used by many laboratories because of the cost and the time, and that is also a time-consuming method. And then the final question is, well, what do we have to do if we are working in networks, and if we have to compare time series or data obtained in different places, in different coastal waters or different oceanic waters? Do we have to use the same methodology? or do we have to use all of us HPLC? In general, it is recommended that in these cases, we have to use the low-cost methods, spectrophotometers or fluorometers, and of course, considering your range of variability in chlorophyll-a concentration. If my study place or study site is characterized by high chlorophyll-a concentration, well, I can use a spectrophotometer without any problem. But if I am working with very lithotrophic water, I will have to use at least a fluorometer. Then, if we are using an HPLC, it is recommended that in order to be comparable or data be comparable, it is necessary to use the same method, for example, Thomas method. And finally, it is very recommended intercalibration exercise to evaluate how comparable are the data we are generating as a network. For example, the sea hair intercalibration round robin is an example of an intercalibration exercise. We have to think about if you are working in a network to make some similar type of exercises. And I recommend you to take a look on the publication of Farjon and McLean from 2002. They talk about the source of variability in chlorophyll analysis by fluorometry and HPLC, also in a Symbios intercalibration exercise. In this case, they focused on fluorometry and HPLC. They did not include spectrophotometry, but it can be also added if we want to integrate all the type of measurements we are doing as a network. Well, finally, I want to mention that many of the information I considered in this presentation, it was based on the book by Jeffrey of 1997, Phytoplankton Pigments in Oceanography, Guidelines to Modern Methods. In particular, this book focuses on HPLC, but it also makes an excellent review of the other methods. It is free in the network. I am including here below the page where we can download the book and to consult. And that is all. And thank you very much for your attention.
Thank you, Adriana. Really interesting. I was wondering if I can start the question, opening questions. So do we have an idea on our project, on the methods that our participants use, if they are similar?
In particular, in the Latin American stations, we are using spectrophotometry and fluorometry. It changes according to the station. In Ensenada, we are the only one that have an HPLC. We are using the Thomas method, indeed, but we also use spectrophotometers to have a comparison among them. And we are having good results because we are working with coastal waters. But yes, the problem is if you have to go to offshore, the spectrophotometer is not appropriate.
So then it should be a fluorometer in case I don't have 100,000. Yeah, okay. Thank you. So we are opening for questions. You feel free to switch on your microphone, introduce yourself, your name, where you're from, or you're free also to make your question on the chat. the chat window, and I can read it out loud. Um, Edwin Pinto says, please would you send us a PPT about chlorophyll? So Edwin, this Dr. Gonzalez presentation, the PDF will be available in the NANO website. Are you a NANO member? If not, please leave me your email address. You can send me a private message and I can email it to you, okay? So Shubrata,
Yeah,
hi, how are you?
I'm fine. And you?
Good, thank you. So yeah, your
Yeah,
question?
yeah. Thank you, Andrea, for your nice presentation. And I am Shubroto Sarkar from Bangladesh. So I have actually two questions. One question, first question is about the sample collection. So let's say if I have a station which is far from the lab, and if I don't have any facilities to filter the water sample in the field, then what should I do?
Okay, I suppose you don't have liquid nitrogen,
Yeah, I
but
would say if I don't have any chemicals or
and
filtration
uh-huh.
facilities,
And how long it will take to you to go from your study site to your laboratory?
6 to 8 hours.
Okay, and yes, you can maintain your samples in a freezer
In the ice
or
box.
an ice box, yes.
Yeah.
And when you arrive to the laboratory, you proceed to the extraction procedure, and that it will be okay. It is
Okay.
recommended that when we use a freezer to store the sample, the filter, after
Yeah.
filtration, up to 24 hours, there is no degradation of the sample.
Okay. Thank you. And my next question is for the least productive area like Bay of Bengal.
Uh-huh.
Should I take larger volume of water? Let's say 2 liters of water.
Well, the best is to filter um, the most amount of water you can,
Mm.
because the sensitivity of the instrument you use will depend on the concentration of chlorophyll in your sample. It minimize errors. For example, for HPLC, it is required 2 liters at least, and for coastal waters, maybe 1 liter or half. It depends on the concentration of pigments you have. I recommend it is if they are oligotrophic waters, 2 liters, yes.
Okay, thank you. And another
Thank
question,
you.
last question.
Yes.
Okay, so at this moment I have the spectrophotometer, but
Uh-huh.
within 2 months I will get the algal torch. So if I collect the water sample from the field and take the bring this sample in the lab, and then I use algal torch in the lab, so is it okay?
I don't understand. I didn't understand which is the one you are acquiring.
So I will have a algal torch in 2 months. So if I bring the sample from the station to my lab, can I use algal torch to analyze the chlorophyll in the lab, or I should? It's better to carry the algal torch in the field.
Okay. Um, Algal torque. I am not familiarized with this.
So it's like BBE,
Okay, okay.
fluorometer.
Okay. I am not sure if you can do it in the field. Well, it will be better because you prevent of any degradation of the sample.
Okay, but if I do it in lab, is it also okay?
Or you can do it in the lab. Yes, if it will not
Yeah,
take so long to be there, the 6 hours you mentioned before, for example.
yeah. Okay. Thank you.
Thank you.
Thank you, Subrata. So we have
Thank you, Lika.
Jonathan is asking, Dr. Adriana, is there any email he can contact you?
Okay, yes, I am reading that. It is Adriana. I am writing here. I am not sure if you can see that, but it is Adriana.Gonzalez arroba uabc.edu.mx. Here it is. I will add my email with to my presentation. I think that I forgot to do that. And when Lilian upload the presentation, you will also have it there.
Yes, I'm adding my email address here as well and the NANO website link.
Okay.
Cassia has a question. Yes. Hi, Cassia.
Hi, hello.
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 Excellent presentation.
Thank you.
Like it. I work in this field. I use HPLC. I did work in oligotropic and coastal environment and with pigment, and now I'm working with coral pigment, and I will finish my doctorate degree now in March. And after that, I'm interested to reach more to this group and be available to help, because I'm a chemistry and we have a lot of problems with degradation, and when you have to calibrate, you have to consider the polar of Chlorophyll-a and the polar of the column. And if somebody needs to ask something about chemistry of these things, I can help if they want.
Okay, thank you very much. Thank you. You can also then send me an email maybe or annotate here your email, Cassia.
Yes, I will send you an email and I will
Thank you.
put here also.
Thank you.
Muito obrigada, Cássia. Any more questions? I think that's it, Adriana. Thank you very much for your talk. Thank you all for your presence.
Thank you.
Yeah, so Dr. Adriana is adding her email here to everyone. And yes, you can contact us at the NANO website or in our social media. Hopefully by the end of the next week, this presentation and recording will be available in the NANO website and Pogo YouTube channel. And next week, we're going to have another presentation from the University of Baja California, Dr. Eduardo Santamaria. So we will be announcing it at the beginning of next week, hopefully. And I hope to see you all there again. So thank you and have a nice weekend.
Thank you, Lilian. Thank you, everyone. Thank you.
Bye-bye.
Bye-bye.